So it's a great pleasure for me to talk to you about some of the advances that exist now in the field of epilepsy. And these are the advances in what we call modifying approaches for disease-modifying approaches for epilepsy. And here I'll focus specifically on Dravet syndrome, which is the type of epilepsy that we study in my lab. What I will say is that the years ahead are very exciting for the field of epilepsy. And as you could hear from the previous talk, is that we still need to do research and find better way to treat epilepsy, because even with all the treatment modalities that are currently in clinic, we still have some of the patients who are resistant to available approaches. And so the approaches that I'm going to be talking about here is within the realm of gene therapy. And this is not all in clinic yet, and it's all in research. And so let me see if I can see that. So I have some disclosure here for the therapeutic approaches that we are developing. And the outline of my talk is that I will talk about start with an introduction of Dravet syndrome, and I know that not everybody will know about this epilepsy, and move on to the etiology and pathophysiology, and then talk about our hypothesis and how we came up to try to understand this epilepsy. And from there, I'll talk about our approaches into therapy and showing you some evidence at the in vitro level on how we develop these therapeutics, move on to clinical or screening of our vectors in vivo in the animals, and then end up with a conclusion and, of course, some question and answer. So as said, Dravet syndrome is a rare and very devastating form of epilepsy. It's belonged to what is known today as the developmental epileptic encephalopathy. And this is a group of very severe form of epilepsy that start usually in childhood and very resistant to current treatment, and very resistant to treatment, and also very difficult to manage for clinicians. And here we know that its incidence is about 1 in 20 to 1 in 40,000 birth, and it represents about 7% of all severe form of epilepsy, starting before the age of 3. I like showing these pictures of kids who have Dravet syndrome, and most of them are met during some of our meetings at the Dravet Foundation. And this is a good reminder of why we are doing this research. From the genetic point of view, the etiology of Dravet is very well known, and this knowledge comes from a seminal paper from Belgium, where a group led by Peter de Jong found for the first time that there was a mutation in SCN1A that they linked to a disease that they call at that point severe myoclonic epilepsy of infancy, which is today known as Dravet syndrome. And they found out that this mutation is typically heterozygous loss of function mutation in the gene called SCN1A. And this is the gene that encodes the voltage-gated sodium channel in its alpha subunit, coding for the alpha subunit, and this is the part of the channel that make the pore. And so this leads to haploinsufficiency of the channel on the membrane. And you can see that normally you have a certain number of channel expressed on neurons. And with the mutation, you have a reduction in this, and that's what we call the haploinsufficiency. And this is showing you as you go from the gene all the way to the expression of the protein on the membrane in neurons in a form of a graphic. And so we know that this heterozygous loss of function mutation are present in over 80% of the cases of Dravet syndrome. And this is not the full story about what causes Dravet syndrome. We also know that as this represents a large part of the cause of Dravet syndrome, some other genes, such as those that are listed here, I will not go into calling each one of them, are also involved in causing a disease that is known as Dravet-like phenotype or Dravet-like disease. And I'm not going to talk about them today. And from a clinical side, we know that Dravet syndrome has a very nice clinical vignette that was first described by Dr. Dravet, who is in a picture here showing with a red sweater just next to me. And I bring this as a part of just acknowledging her. She has passed away early this year. And she was a really big presence in the field, and she has done a lot for the research and for the patients with this particular epilepsy. And we know then that Dravet begin in the very first year of life with seizures that are usually triggered by elevated body of temperature, and in circumstances like during fever or during just an elevation of temperature on a warm day. And in some cases, even excitement will trigger a seizure in these kids, including even music will do the same. And so these are known as febrile seizures or thermal seizures, since they can occur even in the absence of an infection. And as the disease progresses from that time point, we see a lot of different other type of seizures that appear, and these can be status epilepticus, or a very long or prolonged seizure, and usually defined as more than 5 minutes in a simple definition of status epilepticus, but other seizures occur as well. And we know that beside the seizures, patients with Dravet syndrome develop a lot of comorbidities. These are different conditions that are associated with the seizures, and these include cognitive, motor behavior, and sleep and circadian rhythm disturbances. And so all of these create what is known as a syndrome, because it's a collection of multiple symptoms that actually define the disease. A very important thing, as you heard of here, the speaker before me, Stephanie, was talking about SUDEP, and this particular epilepsy has one of the highest rate of sudden unexpected death in epilepsy. And this is this is an area of a lot of concern for family members of those kids with the epilepsy, as it's on every single day they are concerned that another seizure will occur, and there is a high risk of this type of mortality. And Dravet is resistant to most anti-seizure medication on the market, and there have been a lot of development in this field, and we've had even some anti-seizure medication that were specifically developed for Dravet syndrome, and this happened even in recent years, and we have things like fenfluramine and Epidiolex that came on the market. They were approved by the FDA, and even in the presence of this, we still have some seizures. When you look at patients with this epilepsy, they only have partial alleviation of the symptoms. Their seizures are recurrent, and they have multiple comorbidities still present. The side effects are there, and these are usually due to polytherapy. They are using multiple medications, and they have a reduced quality of life, and the risk of sudden unexpected death is still there. And so for us, this was a very big motivation, and for many people in the field, to continue to try to find a better way to treat Dravet syndrome, and because there is this really huge medical need. And here, I apologize for these slides. You cannot read all the details there, but what I want you to see is that these are different type of medications and even therapeutics that are under development around the world by different groups. Here, I will just show you that at the bottom, these are medications that are still in the preclinical phase, going all the way up to the one marked with the yellow. And as you start going up here in this field, these are some of the therapies that are in clinical phase 1 and phase 2. And up here, these are those that have been approved, such as the Epidiolex and fenfluramine and more, as they are here. They've been approved recently. So this shows you that there is a lot of activity in the field to try to find treatment. And all of these groups that are working on these future therapeutics are using different forms of different methods. And some of the methods that are getting a lot of attraction and a lot of excitement in the field for the physician, for the scientists, and even the patients and their families are those that are using very sophisticated, innovative genetic approaches, as some of what I'm going to talk to you about. The idea for that excitement is because there is a promise that these type of treatments are very precise, as part of the precision therapy, and they can treat the disease going to its root cause and may be able to actually alter the course of the disease, and therefore there will be disease-modifying therapies. And so the work that I'm going to talk to you about here has started out over 20 years ago. And of course, as you can imagine, this is very important. First, we wanted to understand what is the mechanism of this particular epilepsy. And to start with that, we generated a mouse model using genetic approaches where we deleted the part of the gene that encodes the part of the protein highlighted here. And this was not very obvious, but when you do this, we were very surprised to see that the animal will start having seizures and develop multiple symptoms of Dravet syndromes. And so over multiple years, we conducted a series of studies, tried to understand the pathophysiology, what is the mechanism of this epilepsy. And then from there, we had a lot of lessons that we learned. And here, I'm just going to bring two of the key lessons that we learned that are very critical to how we thought about developing these new therapeutics. One is that we understood that Dravet-causing mutations lead to reduction of sodium current, and of course, these are SCN1A mutation. The gene is responsible in building this protein called NaV1.1, or a sodium channel. And so we see with the mutation in this gene, we have reduced sodium channel, we have reduced excitability of inhibitory neurons, but not excitatory neurons. So this was very critical to us, that it was a very specific impact on this type of cells. The second thing that we notice is that key Dravet phenotypes or symptoms are recapitulated when we do an experiment by introducing the mutation specifically in inhibitory neurons. This told us that having the mutation only in these cells was sufficient to cause the disease, and therefore these cells are key driver of the disease. And because of that, we thought we came up with what is known today as the interneuron hypothesis of Dravet syndrome pathophysiology. And in simple words, what you can think of is that Dravet is a disease of forebrain interneurons, because here we are delivering this, the gene only in the forebrain interneuron. And so with that understanding, before I go to the next point, I should say that that was our hypothesis, but today we know that a lot of other groups have been working in this field, and they understood that not all interneurons participate the same way in causing the disease. It's principally what are known as parvalbumin interneurons that are key driver of the disease. The other thing that has been understood in the field by others is that there is a transient period during development where the inhibitory neurons are the key participant or key driver of the disease, but after you pass a certain age, some other things happen, and those like excitatory neurons and some synaptic process become part of the pathophysiology of the syndrome. But with going back to our hypothesis of interneuron being the key driver overall, we thought that if we make a therapy that targets these interneurons, we'll be able to efficaciously prevent Dravet. And this may occur with very little side effect because we know exactly where things are coming from, and we can do a precision therapy of that sort. And that was really good for us to think of, but the key problem that we had at that point is that the science was not at that level to be applied. We needed to develop two things, and that's because there were two challenges that we needed to overcome. One is that when you want to deliver a gene therapy, you use AAVs, and these are adeno-associated virus, that they are the carriers of choice, and especially when you are working on neurological diseases. And the problem is that AAVs capacity is, or packaging capacity is only 4.7 kb. Whereas the size of SCN1A is 6 kb, and making it too big to fit into the AAV as a carrier. And the second thing that we came up across is that we wanted to target inhibitory neurons because we found that this disease has a cell-specific etiology. The inhibitory neurons were the key cell population to target. But the problem with this was that we did not have ways to go and be very precise in targeting interneurons. The way you do it in biology and in science is that you use some element called promoters or enhancers that will go and deliver the gene only into the cells of interest. And those promoters for inhibitory neurons, at that point, we had only one that was very reliable, that's GAD2, and this one was already too large to fit into the package, the AAV cargo space. And then we couldn't use it, and thinking that it's already too big for the cargo space, and thinking of using it even in combination with actually the treatment component of the cargo. So all of that I just said can be summarized in this analogy. We had a large molecular machinery that we wanted to deliver to help treat this epilepsy. And we had a small vehicle, we had a small truck, and our GPS was very defective. It couldn't take us to the inhibitory neurons where we wanted to go within this very complex architecture of the brain. So what do we do? So we collaborated with our friends at the Allen Institute, and where they were working on a project where they wanted to find this enhancer using RNA-seq. They find this little enhancer, discovered a new element, basically kind of the GPS element that will help them target different cell types, and they will create this nice map where they can see where the different cell types are in the brain and where they connect with other neuronal population within the brain. And so among them, there was one enhancer called the DLX56 enhancer that was the one to target inhibitory neurons, that was working well to target inhibitory neurons. And so we can see that this enhancer can target inhibitory neurons that are labeled in green, labeled in red, sorry, and then you deliver a green protein showing in green here. And when the two overlap, like in these two cases, you see that the cell turn yellow, telling us that there is successful delivery of the YFP or the green protein within these cells. So that was exciting. However, the efficiency was lower because you can see this cell here remained green, did not receive, this cell is green, but was not, did not have the red, meaning that it was not an inhibitory neurons where we are delivering the green protein. So the efficiency was not very good there, so we work with them to try to improve this. Another thing is that we also knew from another group, the Fischel lab, where they are doing developmental biology, trying to understand where are these, how does the brain build up itself, and more specifically, how inhibitory neurons are born, and how do they come to build the whole inhibitory neuron system in the brain through development. And here you can see the same kind of approach where they have inhibitory neurons marked with the red protein, and they deliver the green protein here with the DLX enhancer, and then they see that they can hit a lot of them. You see a lot of yellow, but there are some miss as well there. So we work to improve this approach, and we thought that would be something that we can use for therapy. The next thing that we wanted to do is that how do we deliver this large gene that we wanted to send in to treat Dravet? For that, we rely on another technology called the INTIN technology. This is based on something that occurs in nature for small organisms like some bacteria and fungi and even small plant. What they do is that when they build their large protein, they start out with two different genes. They don't do it with only one gene. They start out with two different genes, and they build half of the protein with small element attached to them called the INTIN. And this little element will come together and join the two big pieces and form the full-length protein. And then excise themselves out of the way. They did not need any enzyme to initiate that reaction, and so they work on their own spontaneously. So this was very interesting to us, and we thought that we can use this system in what in the lab we call affectiously the AKIA method. Basically, we are thinking of cutting the big gene, the intact gene that we want to send in to repair the brain or to rescue the disease. And we cut it in half, package them in two different AAVs, and deliver them into the cells of interest. That was our idea. And then once we deliver them into the cell of interest, they will make their protein with the INTIN segment attached to them, and the INTIN will come together and give us the full-length sodium channel. And we think that if we're successful in doing this, we should be able to treat Dravet. But this is the concept, and now let's go to how we did it in the lab. So we started in the lab in the dish where we build the two constructs, so the two segments of the artificial gene segment, and we attach this CMV promoter, and this is a promoter that allows us to use these HEC cells, HEC293 cells. These are cells that we use in the lab to try to study protein expression. And so we would then deliver these gene segment and recorded electrical activity from them. And this is electrical activity. You can see in this case, this is a full length where we did not cut the gene at all. And we can see that we can record a sodium current here in green. And when we had the two component, we can see that we can also have the two, we can have a large current here. But when we have only one of those components expressing, no current could be recorded there. And here is the statistics. You see a large current for the full length, which is our positive control. And then when we have the two components, we have a large current, but for the other conditions where we had only one component, nothing was occurring. So this tells us that our molecular build, these gene elements that we built into in the dish can come together when you put them in a cell. They can pass the current as they're supposed to. So there is hope that this will create a good treatment. Then we move on and say, what about the looking at the brain? Can we deliver this into the cells of interest? So we did the experiment where we put now these two components into two different AAVs, the carriers. Now they are small enough to go in. They have an enhancer to send them to the inhibitory neurons. And this is a picture of the mouse brain where you can see here in dark colors where we see the expression of the two components. And then here, when we zoom in at the cellular level, I'm going to start on this side. This is an experimental condition where we express the two components, and we could see now with an antibody that will pick up a tag that is attached to component 1, which is on what we call the end terminus of or end component of the gene, and so we can see that in green. And flag, we can see that in red. So we see that we have those two components expressing. And this is where we see the inhibitory neurons. And when we combine, we merge these images, we see that all the colors overlap, telling us that component 1 and component 2 are expressing in inhibitory neurons. And when we had only one component, we see that this tag that's supposed to pick up one of the components is showing nothing. But this component that is present is showing, and it's also in inhibitory neurons. The same thing for the other component, only the green component is showing, and it's also overlapping with the blue inhibitory neurons. So that was good at the cellular level. Then we look at the different regions of the brain, and we say, is that true that these are going in inhibitory neurons? And